Have you ever thought about how just one tiny change in our DNA, like swapping one letter, can sometimes have zero effect?
Right.
But other times, it can completely mess things up, cause really serious diseases. It's kind of wild.
It really is. And we're talking billions of potential changes. So figuring out which specific ones are actually dangerous, well, that's a huge job in genomics.
Yeah, a massive haystack problem.
Totally. Especially with these things called missense mutations. That's where one amino acid gets swapped for another in a protein.
Proteins being the little machines doing work in our cells.
Exactly. And, changing one part might break the machine, but usually it's fine. Actually, over 99% of these changes we see are super rare
Oh, yeah.
and seem harmless.
Huh. Over 99%.
Yeah. So the real challenge is how do we spot the very few rare ones that do cause trouble
Yeah.
and do it quickly?
Okay, so that's the core problem we're tackling today. And there's this new tool, PRESCOTT. That's PRESCOTT.
Hmm.
It stands for Population Aware Epistatic and Structural Model of Mutational Effects. Quite a name.
It is, yeah. Bit of mouthful.
So our mission for this deep dive is pretty clear. Figure out what PRESCOTT is, how it works, and why it's considered a big deal for understanding these genetic variations. And we're pulling this from the research paper that introduced it.
Right. And, scientists have been building tools for this for a while. Some older methods looked at how proteins change across species over millions of years. That gives clues about important parts.
The evolutionary angle.
Exactly. And more recently, there's been a lot of AI deep learning approaches, which are powerful, but sometimes we need methods that are not just accurate, but also faster and maybe easier to understand why they make a certain prediction.
Okay, so where does PRESCOTT fit in?
Well, PRESCOTT actually builds on an earlier model. It's called PRESCOTT.
Right, PRESCOTT first. What's the idea there?
So PRESCOTT, that's the epistatic and structural model. It looks at mutations from three sort of different perspectives, or scales, as they call it.
3 scales. Okay, what's the first?
The first is evolutionary. Think about a protein's history across different species. If a specific spot, an amino acid position, has stayed the same for ages through lots of evolution,
It's probably important.
bingo.
Yep.
It's likely doing something critical. PRESCOTT measures this using a score called TJET. It looks at how conserved that spot is locally within the evolutionary tree. High TJET means basically warning, this part is crucial. Mutations here could be bad.
Okay, evolution points to sensitive spots. Scale number 2.
That's the molecular or structural scale, the protein's actual 3D shape.
How it folds up.
Exactly. And PRESCOTT looks at two things here. First, how buried or exposed an amino acid is.
Mm-hmm.
Is it packed deep inside the core, or is it sticking out on the surface?
Like internal support versus external decoration.
Pretty much. Changing a core piece can destabilize the whole structure. PRESCOTT uses something called circular variance, or CV, to quantify this packing density.
CV for packing. Got it. And the second structural thing?
That involves the surface residues, the ones sticking out. PRESCOTT looks at their physicochemical properties, like charge, size, whether they like water or not.
Okay. Why does that matter?
Because those surface bits often interact with other molecules. Changing their properties can mess up vital connections, like changing the shape of a key so it doesn't fit the lock anymore. That's the PC score.
Right, disrupting interactions. So PRESCOTT uses TJT from evolution and CV and PC from structure. How does it put them together?
It combines them into something called a max score. This gives an overall sense of how critical that position is. But it's clever. It handles structured regions like stable helices differently than floppy coiled regions.
Oh, so?
In those more flexible coiled bits, it tends to rely more heavily just on the evolutionary score, the TJET. Maybe because the precise structure is less defined or less critical there.
Interesting. And you mentioned epistatic in the PRESCOTT name. What does that add?
Ah, yes, epistasis. That's the idea that the effect of one mutation can depend on other mutations already present.
Okay. Like context matters.
Exactly. PRESCOTT tries to estimate this. It looks back through evolution at related proteins in other species and asks, how many other changes seem to be needed for a similar mutation to be accepted or tolerated in those species?
So if a change usually only happens when lots of other changes happen too.
Then it suggests that mutation might be tricky, maybe harmful, if it occurs on its own in our protein's context. It's a way of estimating the historical evolutionary context.
Okay, that's PRESCOTT, evolution, structure, epistasis. So now PRESCOTT. How does it become population aware?
Right. PRESCOTT takes all that information from PRESCOTT and adds another crucial layer, data from actual human population.
Using that huge database, NOMAD.
Precisely. NOMAD has genetic data from hundreds of thousands of people across different global populations, Prescott integrates allele frequencies from Nomad.
Allele frequency meaning how often a specific genetic variant appears.
Yes, and Prescott uses a fairly simple, intuitive idea here.
Which is?
If a specific mutation is found relatively often in healthy people, it's probably not causing a severe disease.
Makes sense. If it were really bad, it wouldn't spread much.
Exactly.
Okay.
So Prescott takes the score, Prescott calculated for a mutation, and then it adjusts that score based on how common the mutation is in Nomad.
It lowers the score if the mutation is common.
Essentially, yes. It uses a specific frequency cutoff and a scaling factor. If a variant pops up more often than a certain threshold, Prescott reduces the initial Prescott score, making it seem less likely to be pathogenic.
So it's not just filtering out common variants, it's actually modifying the risk prediction.
Correct. It paid special attention to variants that are sort of in the middle ground, not super common, but not vanishingly rare either. Say, between 0.0005 and 0.0001 frequency. If Prescott flagged one of these as potentially bad, but Nomad shows it circulating at that level, Prescott dials back the warning. It uses the PopMax frequency, the highest frequency seen in any of the Nomad populations.
That seems like a really important refinement, adding real-world population context. How did it perform when they tested it?
It did really well. They tested it against databases like ClinVar, which lists variants with known clinical effects, pathogenic or benign.
Right, the ground truth, more or less.
Yeah, and Prescott showed a significantly better ability to distinguish the bad from the benign compared to Prescott alone. It also outperformed other well-known prediction methods in that comparison.
So adding the population frequency data really boosted the accuracy.
Definitely. It also performed well on the ACMG dataset. These are genes clinicians look at for actionable findings. And they also compared Prescott, the base model, against results from lab experiments, what are called deep mutational scans, using the protein gym dataset.
How did Prescott do there?
Prescott itself was pretty good at identifying sensitive regions in proteins, areas where mutations are more likely to mess up function. They showed examples like the p53 tumor suppressor protein. Its performance in pinpointing these critical zones was actually comparable to some big AI models like AlphaMissense.
That's impressive for a model that also incorporates structure more directly.
It is. Though interestingly, Prescott tends to be more accurate for mutations in the well-structured parts of proteins compared to the flexible coiled regions, and better in regions where the protein structure prediction itself is more confident.
Okay, that makes sense. Any other quirks or limitations?
Well, yes. For certain populations, like Finnish people who have a unique genetic history, some disease-causing variants are actually more common than elsewhere.
So the Prescott adjustment based on frequency might wrongly downgrade a truly pathogenic variant in that specific population.
Potentially, yes. So in cases like that, the researchers recommend maybe using Prescott directly, without the population frequency adjustment.
Right, context matters again.
Also, for some types of mutations, particularly gain-of-function ones, where a mutation makes a protein overactive instead of breaking it, sometimes the purely evolutionary methods, like an earlier version called GEMI, might perform better. Structure doesn't always help predict those.
Okay, so it's not a one-size-fits-all, but Prescott is generally the go-to.
Generally, yes, especially when you have reliable population frequency data.
And they made this accessible, right? There's a web server.
Yes, which is great. They have an online platform with Prescott, Prescott, and an Prescott database. Researchers, clinicians, anyone can use it.
What can you do there?
You can look up pre-calculated scores for, I think, over 19,000 human proteins. Or you can input your own mutation data, even provide your own population frequencies if you have them, like from the newer Nomad V4.
And it gives you the pathogenicity score?
Yep, the Prescott score, the Prescott score, and even the underlying components like TJET, CV, and PC. You can download everything. It's quite transparent.
So summing up, Prescott is a powerful tool because it integrates these different levels, evolution, structure, and now population data.
Exactly. It combines these distinct biological insights to give a more nuanced and generally more accurate prediction of whether a missense mutation is likely to be harmful.
It's like a shortcut to getting really well informed about a mutation's potential impact, helping to flag the ones that need a closer look.
Precisely. It helps prioritize which variants warrant further investigation, whether in the lab or in a clinical context.
So for everyone listening, here's something to think about. Imagine we could accurately predict the effect of any genetic mutation. Just reliably know what it does. How would that change medicine? Our understanding of health, disease, maybe even aging?
It's a profound question. The implications are potentially enormous.
Absolutely. And tools like Prescott are stepping stones on that path. If you're curious, definitely check out the Prescott web server. Maybe look up a gene you're interested in.
It's a great way to see the science in action.